Now let's go over cell trafficking, or how proteins are sent to one place or another in the cell. The main character here is the Golgi apparatus, which is kind of like the specialized endoplasmic reticulum, which has some modifications for packaging molecules before they're exported to their final destination. This is a scanning electron micrograph of a Golgi apparatus. Honestly, you'll probably never need to be able to recognize this on your exam, but just in case, you can see here that the Golgi is pretty easy to recognize. It basically looks like a cross-section through a stack of pancakes, and sometimes you can see these vesicles coming off of it. As I mentioned earlier, end-linked glycosylation occurs in the rough ER. However, these end-linked oligosaccharides can be further modified in the Golgi. Also, O-linked glycosylation occurs exclusively in the Golgi, and only occurs on serine and threonine residues. So what's O-linked glycosylation? It's pretty similar to N-linked, except now we're adding the oligosaccharides to an oxygen atom, which is on a serine or a threonine amino acid, which is why it's called O-linked. So like before, these are the amine and carboxyl groups that all amino acids have, and here are the oxygen atoms that the O-linked glycosylation will add sugars to. So like I said before, the Golgi is mostly known as the distribution center of the cell. Now how does it know what needs to be sent where? Well, specific modifications on proteins can target them to different organelles, and the most important example of this for your exam is that adding mannose-6-phosphate to a protein targets it to the lysosome. So here's mannose-6-phosphate, which is just a mannose sugar with a phosphate group on its 6th carbon. Now if someone is unable to add mannose-6-phosphate to a protein, where are those proteins going to end up instead of the lysosome? Well, they'll be secreted outside the cell, and this results in a disease called eye cell disease. How can you diagnose this? Well, if you see high levels of lysosomal enzymes in the plasma, that's a big clue. Patients with eye cell disease will often have coarse facial features, clouded corneas, and restricted joint movement, and this is usually fatal in childhood. This might show up on a test as a really sick baby with lab results showing lysosomal enzymes in the baby's blood, and they ask you which of the following mechanisms is responsible for the disease. And of course you'll pick the one about not being able to add a sugar to a protein. Now not only can the Golgi add sugars to proteins, but it's also important for assembling proteoglycans, which are proteins that have lots of polysaccharide chains attached to them. These are often secreted into the extracellular matrix, and used as a cushioning filler substance. So where do you think you might find it? Well, there's lots of proteoglycans in cartilage and in joints, and one of the main ones is chondroitin sulfate, which as you can tell from the chondro in the name, like chondrocyte, is an important part of cartilage. And people even take this as a dietary supplement for osteoarthritis, although I think the jury is still out on whether or not it actually helps. Lastly, the Golgi is where many post-translational modifications are added to both proteins and proteoglycans, such as sulfation of tyrosines on some proteins. So now let's talk a bit more about vesicular transport to and from the Golgi. The Golgi is found between the rough ER and the cell membrane. And the cis Golgi is what we call the side close to the rough ER, and the trans Golgi is what we call the side close to the plasma membrane. There are three main proteins that organize traffic to and from the Golgi, as well as between the different layers. COP1, which stands for coat protein 1, is involved in retrograde transport from the Golgi back towards the ER. It's also used to bring things from the trans Golgi back towards the cis Golgi. So basically everything in this downward direction. Now COP2, or coat protein 2, does just the opposite. This causes anterograde transport from the rough ER towards the Golgi, although this one does not have a role in transport from one part of the Golgi to the other, like COP1 did. Now you might have your own way of remembering these, but I like to think of it as 1 being symbolic of the beginning, like the starting point, so COP1 goes towards the beginning, which in this case is the rough ER, and COP2 goes the other way. Lastly, clathrin is important for receptor-mediated endocytosis, and is also used to send vesicles to lysosomes. When something is endocytosed from the extracellular environment, it's first stored and handled in a temporary organelle called an endosome. Depending on what the endosome contains, it can be sent to a lysosome for degradation, or to the Golgi for more processing, or even back to the membrane for excretion. For our next flash quiz, can you tell me where a protein is going to end up if mannose-6-phosphate is attached to it in the Golgi? That would be in the lysosome, and remember, if this mechanism is defective, you can have eye cell disease.